11 o'clock here, so we're going to get started. Thank you, everyone, for logging in and joining me for today's presentation. My name is Jackie Carville. I'm the Marketing Director here at ImmunoChemistry Technologies. You may have noticed that your phone has been muted. However, I still encourage you to ask questions along the way. Just type your questions into the chat dialog at the left side of your screen, and I will answer them at the end of the webinar. If I can't get to your question during today's presentation, or if your question requires a more detailed technical follow-up or response, you will receive a personal email response right after the webinar. This webinar is being recorded and will be available for viewing in the next day or so. So don't worry if you miss anything, that'll be sent to you. But to get started today, I'm going to be going over a variety of powerful tools for in vitro fluorescent imaging, how they work, and their relevance to cancer research. So, from the get-go, we've got a lot of different tools to cover today, so we're going to be covering them at a pretty high level. Feel free to ask questions, get more detailed. We can get those questions in, or respond to you afterwards. And to keep in mind, we also have hour-length webinars going into more detail on these product lines as well. So in reference to the cancer topic today, we did just get back from the AACR conference a couple weeks ago and had lots of excellent conversations there with researchers conducting this type of work. So if you're joining us today after talking with us at the AACR conference, it was great seeing you there. So as a little background on ImmunoChemistry Technologies, we offer quality biomedical research reagents, including unique apoptosis assays and proprietary immunoassay reagents. We do serve researchers all over the world, having a wide distributor network there, and are really dedicated to helping our customers achieve great results in their research. We were founded in 1994, and we're in Bloomington, Minnesota, which is located just south of Minneapolis in the greater Twin Cities metro area. Originally, ICT was a contract laboratory focusing on custom immunoassay development work and ELISA reagents, but we have expanded and diversified our product lines over time. Now, today we are no longer just a contract laboratory for protein chemistry. We're your trusted supplier for apoptosis assays, ELISA detection reagents, and custom ELISA development and other protein chemistry-related services. Our customers use our products to study human and animal biology, as well as investigate treatments for cancer, degenerative diseases, and other conditions affecting both humans and animals. As a note, all of the products and services that we produce and are going to talk about here today are designed for research use only. So first, I'll talk a little bit about our Eliza Solutions products, which are designed to really help you build a better assay. Our products address a lot of the issues that typically occur during ELISA development, such as specificity, sensitivity, reproducibility, and shelf life. So to address this, ICT coding buffers, blockers, sample and assay diluents, conjugate stabilizers, and wash buffer, they all work together to minimize the buildup of unwanted proteins and generate a very clean signal for you. And in this area, we focus on providing several key ways to optimize your ELISA assays. First, these products minimize your background and enhance signal-to-noise ratios for improved assay sensitivity by promoting a strong signal while also minimizing nonspecific binding and background noise. Next, we can also improve your precision and reduce plate-to-plate variability through the use of our stabilizing buffers, which allow you to prepare plates in batches to use over time, really increasing consistency and providing improved plate-to-plate precision over extensive storage periods. Next, just simply purchasing consistently through a reliable source like us who have been around for over 20 years can also help increase your assay reproducibility as well. Additionally, you can promote a higher specific signal by minimizing nonspecific binding through our specialized blocking and stabilizer formulations. And speaking more to stability, you can also stabilize your protein conjugates through the use of alkaline phosphatase and horseradish peroxidase conjugate stabilizers. And finally, to increase the shelf life of your prepared microtiter plates, by correctly stabilizing adsorbed proteins with reliable coating and blocking buffers. So this will result in plates that can be stored for several months or even years under the proper conditions. So to really summarize all of these points, ELISA development is a long-standing core specialty of ours, and we're happy to work with you to help optimize your immunoassays. So as I mentioned, in addition to our Eliza Solutions, we also offer a wide range of various custom contract services. With regards to our ELISA services, if you're like a lot of scientists, you really just want to focus on your field of study, and you might not necessarily have the background or time to spend it developing a rugged immunoassay test yourself. So pre-developed ELISAs and other assays might be easy to run, but the development of a custom one does require lots of knowledge in immunology and protein chemistry to understand antibody specificity and binding interactions. And that's really where our team comes in. And our scientists are experts with many years of experience in immunoassay development and can also perform optimization and testing services for you too. Outside of immunoassay development, we also provide custom protein lyophilization services. So if you need a reagent vialed and freeze-dried, we can reliably prepare that for you. And most of our lyophilization projects are finished within a week, so we've got some pretty quick turnaround time on that too. So with that being said, I'm going to move on to our fluorescent probe product lines, where I'm going to be focused on for the rest of the presentation today. So our fluorescent cell-based assays are for apoptosis detection, a form of cell suicide mediated by a cascade of proteolytic enzymes known as cysteine proteases or caspases. So these caspase assays, live dead stains, and mitochondrial permeability assays are really essential in the investigation of cellular apoptosis and cell death. So what I'm going to go through here are several different fluorescent probe product lines. Flicka for the detection of active caspases, Magic Red for detection of caspases and cathepsins in real time, MightOPT to assess mitochondrial health, cytotoxicity tests, Fliss for serine protease detection, and then I'll just briefly touch on a new cell viability assay that we have, as well as some oxidative stress assays as well. So first, I'm going to get started by talking about kind of our bread and butter product line here at ICT. Some of our most popular products here are our Flicka Kits. So Flicka stands for Fluorescent Labeled Inhibitor of Caspases and measures cell death by detecting intracellular caspase activity and membrane permeability in whole cells. We do offer these kits in 3 different color options, green, red, and far red. These kits, as well as many of our other assay kits we'll discuss today, are offered in various sizes with different numbers of tests. So we do have some of these trial sizes available for testing out these products. So in terms of cancer research, there are several advantages to using Flicka. First, Flicka is the most accurate and sensitive method of apoptosis detection, as it's measuring the actual intracellular process of apoptosis via caspase activity, instead of a side effect like phosphatidylserine. Second, it's the only method that can detect cells in early apoptosis and differentiate them from cells in late apoptosis as well. And this means that you can see how your cells are responding to treatment, whether they're apoptotic, necrotic, or unaffected. And finally, we have a few different types of Flicka kits available depending on your particular cancer research goals. So to assess apoptosis in general, we have Flicka polycaspase assay kits. But if you'd like to look at a specific caspase, we also have Flicka kits for caspase 1, 2, 3 and 7, 6, 8, 9, 10, or 13. So if your project's looking at one of those in particular, we have those special kits available. So how does Flicka work? Our Flicka probes are comprised of a caspase affinity peptide sequence, floral methylketone moiety, that facilitates an irreversible binding event with the targeted active caspase enzyme, as well as a fluorescent tag reporter. Flicka is a cell-permeable direct probe, and that means that the cell does not require any lysing, permeabilization, additional reagents, or antibodies for detection. To use Flicka, all you have to do is add the non-cytotoxic Flicka probe to the cell culture media. Once Flicka is diffused into the cells, it will interact with the enzymatic reactive center of a targeted activated caspase and then form an irreversible covalent bond, marking the cell with its fluorescent signal. There's no interference from inactive forms of the enzyme throughout this particular process. Unbound Flicka reagent is then washed away, and the remaining fluorescent signal is a direct measure of the caspase activity present at the time of Flicka addition. You can then visualize your cells using a fluorescence microscope, or if you'd like to quantify the fluorescence intensity, you can use a fluorescence plate reader or flow cytometer as well. Our green fluorescent FAM Flicka probes, those will excite at 488 nanometers and emit at 530. The red fluorescent SR Flicka probes will excite at 565 and emit at 595. And our far red fluorescent Flicka 660 probes will correspondingly excite at 660 and then emit at 690. So those are kind of the three different options we have for Flicka depending on your technology that you'd like to use for analysis. Now that we know how that process works, we can check out some example results, starting with the flow cytometer data here. And what you're looking at here are results from three different Flicka products. So first is our 660 Flicka polycaspase product, which is a far red fluorescent probe. And you can see the Jurkat cells are treated with stereosporin, a chemical known to induce apoptosis, so that makes for a great positive control here. The red line is untreated cells, and the black line is the treated cells. And as you can see across all of these products, there's a strong shift in terms of fluorescence intensity among the treated cells. The middle graph is showing a green fluorescence Flicka kit, and the end graph is showing another far red Flicka kit, but this time measuring pyroptosis instead of apoptosis. So next we're looking at here some sample plate reader results. And again, we're looking at Jurkat cells, which were treated with stereosporin, and then read on the plate reader with excitation and emission at 550 and 590. Here we were staining with our polycaspase detection kit, and again, in our induced cell population, there's a very strong increase in fluorescent signal. And finally, here you're looking at some sample fluorescent microscopy results with Flicka. On the left, you can see THP1 cells that are duly stained with our FAM Flicka polycaspase assay kit, as well as hooks to stain the DNA in the cells blue. And if we look at this image, we can see three different cells. The one in the middle is the one we believe to be apoptotic, as we can see both green fluorescence for the caspase activity. We also see some bright blue spots in this cell, indicating that the cell is beginning to die as the DNA is fragmenting. The cell in the upper left corner is necrotic, as we see no green and we do see scattered blue. And then finally, the cell in the lower right corner is live and non-apoptotic with a lack of green fluorescence, and we can see that one really bright concentrated blue spot that represents the DNA here. The second image on the slide on the right is of colorectal epithelial cells that were infected with salmonella. And here the authors used a salmonella strain that's constitutively expressing M. cherry, so the salmonella appears red in this particular image. The authors then used our FAM Flicka caspase 1 assay to look for the activation of pyroptosis, so that will stain green. And you can see that these cells in the middle are very heavily infected with salmonella and also have a strong activation of caspase 1. So I'd like to go ahead and move on now to talk about our line of Magic Red assays. And Magic Red reagents are cell-permeant fluorescent substrate probes used to quantitate caspase or cathepsin activity in cultured cells. And these Magic Red kits allow you to monitor your caspase or cathepsin activity in real time, so you can watch your cultured cells undergo apoptosis. These kits are not ELISAs and do not use any antibodies like Flicka, so instead they use cell-permeant substrates specifically targeted by active enzymes. And we do offer a few different types of Magic Red kits, including those that assess activity of cathepsins B, K, or L, as well as caspases 3 and 7. So in terms of cancer research, elevated cathepsin enzyme activity in serum or the extracellular matrix often signifies a number of pathological conditions. Cathepsin-mediated diseases include Alzheimer's, numerous types of cancer, autoimmune-related diseases like arthritis, and the accelerated breakdown of bone structure often seen with osteoporosis. Specifically, upregulated cathepsin B and L activity has been linked to several types of cancer, including cancer of the colon, pancreas, ovaries, breast, lung, and skin. Upregulation of cathepsin K has been shown in lung tumors, and increased cathepsin K activity has also been shown to be linked to degenerative bone diseases, including osteoporosis and postmenopausal osteoporosis. So in general, assessment of elevated levels of extracellular cathepsins has proven to be a very important prognostic indicator of cancer chemotherapy outcome. So Magic Red works when the target substrate peptide sequence is linked to a red fluorophore. Magic Red, also known as Crescel Violet, fluoresces red once the substrate is cleaved by the specific enzyme. So, with our kits, that specific enzyme would be caspases 3 and 7 or cathepsins B, K, or L. If it is cleaved by an active protease, the released Magic Red fluorophore will fluoresce, stay inside the cell, and then often aggregates inside lysosomes. The cathepsin enzymes are lysosomal, while the caspases are not. As protease activity progresses and more substrate is cleaved, the red fluorescent signal increases, so you can actually watch the color develop over time. These reactions are very fast. They start within 15 minutes, and you can watch the fluorescence develop over several hours. The typical incubation time for people running these assays is only about 15 to 20 minutes. So after that, you can watch the cell through a microscope and take pictures, or read total fluorescence of the sample with a fluorescence plate reader. And Magic Red will excite from 540 to 590 nanometers and emits at greater than 610 nanometers. So now we're going to go through a few examples of different Magic Red results. So on this slide, we can see an example of what a Magic Red time lapse would look like. Here, rat fibroblasts were used with the Magic Red caspase 3 and 7 kit. The red fluorescence becomes brighter as caspase activity and apoptosis progressed. And we do have a nice video of this on our website if you'd like to go see that in live action. Next, here's an example of a positive cathepsin B Magic Red assay in THP1 cells. And you can see really well in this picture the cathepsin B concentrating in the lysosomes that we talked about earlier. And finally, this image shows dual staining of camptothecin-induced MCF7 breast cancer cells using both Magic Red and Hookst. So experimental cells were stained with Magic Red for about 30 minutes, washed twice, and then stained with Hookst for about 10 more minutes. And you can see here the apoptotic cells bearing orange lysosomal bodies with the less intense blue nuclei. The non-apoptotic cells have very bright blue nuclei and absent or very much reduced orange lysosomal staining. And so we can see that in this particular experiment, the treatment that was being applied here is killing the positive cells. I'm going to move on now to our MightOPT kits. These allow you to quantitate mitochondrial functionality and apoptosis. Detection of the mitochondrial permeability transition event provides an early indication of the initiation of cellular apoptosis. And this process is typically defined as a collapse in the electrochemical gradient across the mitochondrial membrane. So these kits will detect a change in mitochondrial membrane potential. These kits are very fast, incubation only typically being about 15 to 20 minutes. And we do offer 3 different types of MightOPT kits, and I'll talk about the difference between these 3 in a minute here. But first, looking at mitochondrial health is important because depolarization of the inner mitochondrial membrane can lead to an opening of the mitochondrial permeability transition pore, resulting in the leakage of intermembrane proteins that facilitate apoptosis. Because mitochondrial dysfunction has been closely linked to such neurodegenerative diseases such as Alzheimer's, Parkinson's, and lateral sclerosis, mitochondria do remain a very important organelle of study. So as I mentioned, we do have some different types of MightOPT assays which work in slightly different ways. So first, I'm going to go through how our MightOPT JC1 assays work. And these JC1 assays were our original MightOPT reagents. They're based off a fluorescent cationic dye, commonly known as JC1. And after you add your MightOPT JC1 reagent to the media, it easily penetrates the cells and enters the healthy, negatively charged mitochondria, where it aggregates and fluoresces orange-red. As the mitochondrial membrane potential collapses, MightOPT JC1 is diffused throughout the cell. And once dispersed, the reagent assumes a monomeric form and fluoresces green. So this can be read with a fluorescence plate reader, flow cytometer, or fluorescence microscope. So here, Jurkat cells were treated with sterosporin for 2 hours to induce apoptosis. Cells were then stained with MightOPT JC1 for about 20 minutes and washed. And if we look at this data image, we can see that the 2 cells on the left are healthy. You can see the mitochondria with polarized inner membranes fluorescing red. And on the right, in contrast, we see 3 cells that are apoptotic. And here the mitochondria are at various stages of permeability, so they exhibit much reduced red fluorescence and increased green fluorescence as the MightOPT reagent disperses throughout the cell. So our other types of MightOPT assays are our TMRE and TMRM assays. These differ from JC1 in that they are based on different cationic fluorescent redistribution dyes. So tetramethylrhodamine ethyl ester, which is the TMRE, and tetramethylrhodamine methyl ester, which is the TMRM. And similar to what we discussed with the JC1 kits, after the reagent is added, it enters the negatively charged mitochondria, accumulates, and then fluoresces red. When the mitochondrial membrane collapses in apoptotic cells, the TMRE or TMRM dyes can no longer accumulate within the mitochondria, and then the dye disperses throughout the cells. So the overall cellular fluorescence levels will drop dramatically here, and this event can be easily detected by monitoring the amount of emitted fluorescence done through a flow cytometer, plate reader, or fluorescence microscope. So if we look at some sample results here, you can really see the difference between the healthy and apoptotic cells, with those depolarized apoptotic cells exhibiting much lower levels of fluorescence. So I'm shifting gears here to another product line to introduce our cytotoxicity assays. We have now developed 2 different assays to measure cytotoxicity. If you're looking to measure necrotic and membrane-compromised cells, you should opt to use our basic cytotoxicity test. However, if you're looking to further distinguish apoptosis from necrosis, leading to a more accurate overall assessment of cytolytic activity, we do offer a total cytotoxicity test. And at least 10% more cytolytic activity is often measured when apoptotic cells are also identified with the total test kit. And these kits provide a direct measurement of cytotoxicity rather than using an indirect indicator, such as the release of LDH or ATP enzymes. Again, as with our other kits, there is no need to lyse any of your cells for these cytotoxicity studies. Since these assays do not use any radioisotopes such as chromium-51, they are much safer and much more cost-effective than those comparable kits. There's also no waiting with these kits, so you can directly measure cytolytic activity without having to wait for enzyme release. In terms of cancer research, cytolytic activity is a very important process for eliminating intracellular pathogens in cancer cells. This process is accomplished through various immune effector mechanisms, including natural killer leukocytes. Natural killer activity is facilitated by non-specifically lysing infected targets through the use of natural killer receptors, recognizing IgG bound to specific antigens on the target cell surface. Natural killer cells may also induce apoptosis in target cells. The activity of natural killer cells and their effect on target cells is very frequently studied in various immunomodulation experiments. So first, I'm going to run through how our basic cytotoxicity tests work. So first, you purify target cells and stain with CFSE, which is a green membrane stain, to distinguish them from unstained effector cells during flow cytometry analysis. Next, you'll add your effector cells or toxic agent to the target cells, and the effector cells or toxic agent will act on the target cells during this step. Next, you'll add the red live dead stain 7AAD, which will enter cells with compromised membranes in necrosis and late apoptosis and then bind to the cell's DNA. The stain will not enter cells with intact membranes. Finally, you'll run your controls and analyze using flow cytometry to quantify two populations of target cells in each sample, both the live target cells and the necrotic cells. The percentage of cytotoxicity is then calculated by counting the number of dead target cells, which are stained both green and red, and then dividing by the total number of live target cells, which are all the green cells. To then expand on the capabilities of the basic cytotoxicity test, now we'll go ahead and take a look at how the total cytotoxicity test works. The first two steps of this kit are the same, purifying the target cells, staining with CFSE, and then adding the effector cells. Where this protocol starts to differ is that next you'll add the orange-red SR Flicka apoptosis detection reagent. That reagent will enter the cells and bind to active caspases in this step. If the cells are not apoptotic, it will just wash out of the cells. Then again, you'll add the red live dead stain 7AAD, which, just like in the basic kit, will enter the cells with the compromised membranes in necrosis and late apoptosis and bind to the DNA. Finally, you'll run 8 different controls and analyze using flow cytometry to quantify 4 populations of target cells in each sample, including cells in early apoptosis, which are not detectable by any other method. The 4 populations that you'll be looking at here are live target cells, early apoptotic cells, late apoptotic cells, and necrotic cells. So then we're going to spend some time, we'll analyze some flow data for both the basic and total cytotoxicity tests so you can really see the breakdown of these different cell populations of interest. So on the left, we can see flow cytometer data for the basic cytotoxicity test. Here we've created a plot with CFSE versus 7AAD. All of the target cells here are initially stained green, so the entire population of target cells can be counted in R2 plus R1. After the effector cells and the red live dead stain were added, all the dead target cells fluoresced red, which will shift them up the y-axis, clearly distinguishing them from the live target cells. So we can see the dead target cells are specifically in R2 and the live target cells in R1. In this graph, the live effector cells would be in R4, dead effector cells are in R3. And if we were to use the total cytotoxicity test kit, we would further analyze R1 and R2 to isolate apoptotic cells. So now in looking at the plot at the right, that's an expansion of the R1 and R2 quadrants from the plot at the left. Once the green target cells from R1 and R2 have been gated, we derived a plot of the polycaspase apoptosis reagent SR Flicka versus that live dead viability stain 7AAD. Now we can go ahead and analyze our 4 different populations of cells, the live cells and the 3 populations of cells in the death process. You can see in quadrant 1 that there are live cells stained green. Quadrant 2 has early apoptotic cells stained green and orange. Quadrant 3 has late apoptotic cells stained green, red, and orange. And quadrant 4 has necrotic cells stained green and red. And I just want to point out here, too, that quadrant 2 is of particular interest, as no other methods such as PI staining or other assays can identify this particular additional population of cells. So the overall takeaway here is that researchers are now able to include apoptotic cells in their analysis of cytotoxicity, leading to overall more accurate results. So moving on here, now I'd like to go ahead and cover our Fliss product line. The Fliss reagents, or fluorescent labeled inhibitors of serine proteases, are cell-permeant fluorescent inhibitor probes used to quantitate serine protease activity in cultured cells. Our Fliss assays are available in both green and red probes. So serine proteases are defined by the presence of a serine residue at the active center of the enzyme. The most characterized enzymes of this type are trypsin and chymotrypsin. And all living cells have a base level of chymotrypsin-like enzymatic activity, which will vary with the physiological state of the cell, as well as vary by cell type. Activated serine proteases play major roles in several different functions, which include apoptosis, markers of tumor malignancy, diagnostic and prognostic indicators of breast carcinomas and neck and head carcinomas. So serine protease activity is also altered in a variety of other cell-mediated diseases related to transplant rejection and infections. For our Fliss kits, all you have to do is add the reagent directly to the cell culture media, incubate, and then wash. Because ICT's Fliss inhibitors are cell-permeant, they will very efficiently diffuse in and out of all your living cells. If there's an active chymotrypsin-like enzyme inside of the cell, it will covalently bind with the Fliss inhibitor and retain the green or red fluorescent signal within the cell. Cells containing lower concentrations of chymotrypsin-like enzyme activity will retain a much lower level of fluorescence compared to cells containing higher concentrations of this effector enzyme component. There's no interference from proenzymes or inactive forms of the enzymes throughout this particular process. If your treatment is activating chymotrypsin-like serine proteases, your positive cells are going to fluoresce brighter than the normal baseline negative cells, which allows you to very clearly differentiate the populations. So now to tie back to the beginning of the presentation, you can use these Fliss kits in conjunction with our Flicka Kits, which is what you're looking at in this figure. Here, HL60 cells were treated with camptothecin, the apoptosis inducer, for about 3 hours, and then stained with our green FAM-Fliss serine protease inhibitor and ICT's red SR Flicka polycaspase inhibitor for an hour. Cells were then washed and analyzed on a flow cytometer. And so we can see in the flow cytometry data here, the cells with active serine proteases will stain green along the y-axis, and cells with active caspases are going to stain red along the x-axis. And we can look at some images of the cells and see the range from picture A there, only the green cells showing serine protease activity, B, C, and D with dual stain cells, and picture E only stained red for caspase activity. So to move on here, I wanted to briefly touch on a new cell viability assay of ours that's proven to be pretty popular with people. Our Necrosis versus Apoptosis kit allows you to detect both apoptosis-associated cytotoxicity events as well as cell death due to necrosis. This kit utilizes both our Flicka reagent probe to detect the apoptotic cells as well as the 7A-AD vital staining dye binding to GC-rich regions of DNA to detect necrosis or late-stage apoptosis. So ultimately, this kit allows you to really understand the overall health status of your cell population. It's also a very useful tool for researchers who are investigating the effect of their novel drug or therapeutic, so they can assess their experimental outcome as well as evaluate the overall treatment of the cells. If you want more detailed information on this kit, ICT's Dr. Christy Strandberg recently presented a full webinar titled How Healthy is My Cell Culture? This will go in much more detail on this particular product, and this webinar can be viewed on either our YouTube channel as well as on our webinars page. And finally, I'd like to go ahead and wrap up the webinar today by just briefly touching on our oxidative stress kits. And these oxidative stress kits have been a new area of focus for ICT, so we are actively developing and expanding this product line. So if there's anything you're looking for in this particular area, please feel free to reach out to us, let us know, and we're happy to look into that to expand this product line. So currently we offer oxidative stress assay kits to detect glutathione, hydrogen peroxide, and nitric oxide. In addition, we've also very recently launched a DNA damage ELISA kit. This will quantify 8-hydroxy-2-deoxyguanosine. And I also want to mention that coming very soon, we will soon be launching a total ROS kit as well. So if that's something that you're interested in, feel free to ping me and let me know, and I can make sure that you're notified as soon as that kit is launched. I won't expand much on these kits in this particular webinar, as Dr. Christy Strandberg will be presenting a webinar next month going through how these kits work and sharing some data from these kits. So if this is an area of interest for you, I'd encourage you to check back next month and attend that webinar as well. So with that wraps up the variety of assays that I wanted to talk about today. To recap what we all went over, since we covered a lot of ground, we talked through Flicka for detection of active caspases, Magic Red for detection of caspases and cathepsins in real time, MightOPT to assess mitochondrial health, cytotoxicity tests, and Fliss for serine protease detection. We also briefly touched on our apoptosis versus necrosis kit, as well as our offerings for oxidative stress assays. So again, since we covered a lot of material, I did want to point out the variety of resources that we provide. Our ICT webinar series covers a variety of topics. Again, you can register for upcoming webinars as well as watch webinar recordings from previous sessions from our webinars page. We also have some short product videos on our YouTube channel right now that give you a visual overview of how easy these kits are to use, and you can check them out in action, so I encourage you to give those a watch. We also update our blog with helpful articles, tips, and tricks for all things immunoassay related. And finally, we have an extensive variety of technical documents hosted on our website for each of these kits to really support you in your work and give you lots of detail there. So here's a nice picture of our full team here at ICT. We take really great pride in excellent customer and technical support. We offer very fast shipping, and if you have any questions, you can often talk directly with the inventor of the product to help troubleshoot your process. For those international customers, we do have a large distributor network around the globe to assist in getting you our products as fast as possible. We also have a guarantee on all our products, so if for some reason they don't work and we're not able to help you troubleshoot the process, we will refund your purchase, full money-back guarantee there. So that's about all I have today. My contact information is as follows. If you'd like to reach out to me with any questions or comments, if you have any ideas for future webinar topics that you'd like to see, feel free to reach out to me as well. I'm going to go ahead and look through. I've been getting lots of questions in, so I'll go ahead and field some questions. Again, feel free to keep chatting your questions in. If I can't get to them now, in the next couple minutes here that we have left, I'll make sure to follow up directly after the webinar with you. So it looks like we're initially getting in a lot of questions about Flicka in particular, so I'm going to try and address those first in a chunk and then move on to some other products. So I have someone asking just how Flicka is different from various other caspase detection kits. And to answer this, I'd say that our Flicka assays are used with these whole living cells, so again, no lysing or permeabilization necessary there. It's also not an ELISA and does not involve the use of any antibodies. Also, because active caspase enzymes bind to Flicka, there's really no interference from procaspases or inactive forms of the enzyme throughout the process, so that's a good thing. And we also have a lot of flexibility inherent in these kits, as not only are they available in the various colors, but they can also be analyzed by the microscopy, plate reader, or flow cytometer as well. Okay, we have another person asking how long the Flicka assays usually take to run. These Flicka assays, they start to react within about 15 minutes of addition to the cells. The assay can be incubated up to 72 hours without killing any of the cells. Typical incubation periods for customers, though, are about 1 to 4 hours, so you really can start and finish your experiment fully in one day. Big benefit there, obviously. The exact incubation time really is determined by your own experimental protocol, and again, if you want to chat through that, feel free to give us a call, and we're happy to give you some advice on that end as well. We also have another question here referencing Flicka, asking how sensitive is Flicka? And our Flicka kits are extremely sensitive kits. They even detect the natural apoptosis occurring in the control cells, so generally about 2 to 6% of the population. But the positive apoptotic cells will often generate a signal, from 2 to 15 times greater than the control population there. So very, very sensitive kits for your purchase. Okay, we have another person here who's working with frozen cells. So they're interested to see whether some of these reagents might work with their frozen cells. And I would say to that, the Flicka kits, they might work with some frozen cells and very thin frozen sections. The important thing to keep in mind with our reagents is that they require the cell membrane to remain intact, and the freezing process often will create pores in the membranes, which allows the positive fluorescent signal to leave the cell. With regard to the Magic Red kits, those might work best with frozen cells when measuring the total fluorescence with a plate reader. So if that's the approach you're going to take, that might work best when you're working with frozen tissue. Okay, and then, to just take one more question here to keep us under time, we have someone asking about fixing their cells using the MightOPT reagents, whether they can fix the cells before or after. And unfortunately with these kits, no, the fixation methods will definitely cause a disruption in that proton pump-driven membrane potential gradient. This disruption here would cause all fixed cell populations to appear essentially non-fluorescent, leading to the complete invalidation of all data observations. So I would, not recommend fixing the cells for that particular workflow. Okay, I do still have a few questions remaining. What I'm going to do just to wrap up the webinar here is make sure that everyone's followed up with after the webinar, since that's about all the time we have left today. But I do want to thank everyone so much for joining us. As I mentioned, our next webinar is going to be held next month in June on the topic of oxidative stress. So that date and registration link are going to be available very soon. So I would encourage you to check back on our webinars page for that. And again, as I mentioned, the recording for this particular webinar is going to be available within the next day or so. So thank you again, everyone, for joining us, and have a wonderful rest of your day.